Hi everyone, welcome to this week's Club of Med Talk. Today I'm going to be introducing Dr. Megan Sir, who is an assistant professor of biology at Meredith College. She got her PhD in 2019 at North Carolina State University, kind of doing some special work, especially as she was a part of this cohort where she was able to really be a part of IGERT, and she got this minor in this genetic engineering society. And so she really does some interesting work in terms of this pest control that we're going to learn about today, especially for what is considered like rodent gene drives for different aspects of conservation as well as human health. Her talk is really about this evolutionary considerations for that rodent gene drive for conservation and human health, as I just mentioned. And today she's going to go over a couple different things. She's going to talk about some One Health initiatives, the history of kind of like these invasive rodents on islands, as well as the traditional methods of success and failures for these particular types of gene driving. And then she's really going to focus into the gene drive techniques and the work she does on these gene drives and evolution that she's kind of done in her PhD, as well as continuing right now at Meredith College. And so this idea of One Health is pretty important. We're talking about this, not just one type of health. We're talking about human health, animal health, the environment health, and how that comes together and really interacts. And that can be quite important. And a lot of her work is done on these rodent populations or more in this herpetology setting. And it's really important to start kind of knowing the background, particularly for these rodents. And we know that it's going to be pretty important in these island settings. So these islands often have less than 5% of that land area, but they have about 40% of the endangered species. And there have been about 80% extinctions since a pretty long time on these islands. And so we have to consider things like mice. Mice on these islands, they're pretty much invasive rodents. They're going to be threatening that native wildlife, particularly seabirds. And so when we're looking at this, every red dot that you see here is an island that has an invasive house mice. And so these mice can really start to get everywhere on these islands and do some pretty big damage in terms of what they're doing to the habitat or other animals, as well as humans, because there are going to be vectors for disease. Just going forward, just know that there are going to be some sensitive images because of these mice. For example, here we have this bird, this midway atoll, and the house mice that you saw on the first slide that we've been talking about have actually known to start attacking these nesting birds. And we can start to see that these mice are doing major damage to the birds to the point where they're getting really injured and hurt. And so there's this idea where we have to need to start pulling back on these mice, whether we're suppressing their presence or we're eradicating them on these islands. And the traditional methods for these is going to be something like rodenticides and primarily these anticoagulants. These are going to be aerial applications that are typically only on uninhabited islands. These are off-target effects, and there are concerns for animal welfare on the islands as well when these happened. And these have actually primarily been successful for these uninhabited islands, and is pretty much the standard route of procedure when we're looking for these eradications. However, these rodenticides are second-generation anticoagulants, and so these are often laced in bait and spread aerially. But we have to remember that there are other social implications of this, because that is not a, we say, a nice death for these rodents. But the goal here is to eradicate them and suppress them on these islands. But we also have to consider this evolutionary resistance that could be happening eventually these mice will become rodenticide resistant. And so what happens then? Well, we're going to see this higher failure rate for mice than considering rats, mainly because these mice have a lot smaller home ranges. They have this bait palatability issues. They have this evolutionary resistance to the second generation anticoagulants. And one of Megan Surr's colleagues formerly would like to say failure is easy. All it takes is one single pregnant female left behind. Because, you know, these mice reproduce a lot and they can reproduce very quickly. And so it's pretty difficult, if not impossible, to perform eradications with these airily spread rodenticides. And contrary to brown rats, for which only one predominant genotype was found, there are a lot of different single mutations that are observed in house mice. And here we can see some of these SNPs that are found. These are the mutations that are occurring, and we're going to see high variability in these SNPs. And so kind of coming back to like this general picture of this gene drives in this evolutionary field, especially with these rodents, we can start to understand and look at how the DNA of these rodents might be changing and how that could potentially affect onwards into, say, these bird populations that are also now being destroyed. These gene drives is a genetic construct that is preferentially inherited by offspring. And so in this case, we can think of something saying producing a desired phenotype. So whether that be something that is sex-based, so having your population be more male-based instead of female, so there'll be less reproduction, or maybe that phenotype is going to be something like sterility. And this can spread at near 100% ratios. They can be meiotic or gametic driven. These are naturally occurring or synthetic. Megan Surr is going to be talking more about this in a naturally occurring setting. But it is to be reminded that there is no technique that has been yet fully developed or deployed within the field on these islands. And from here, I will let Dr. Megan Surr take it over.
John Godwin, who was my PI, Fred Gould, who I saw, I think, earlier on here, who was the formation of the Genetic Engineering Society Center, and David Threadgill, who was at NC State at the time, met with some folks from Island Conservation. Many of you may know that talking about gene drives has been around in the field, especially in entomology, things like Project Malaria. And the thought was, could this be applied to something like rodents? And is there a need for something like this? And clearly there is by the pictures I showed you. So this is about when I started my PhD back in 2013. I was a cohort of 6 in the newly formed Genetic Engineering Society Center. And this was an IGERT, Integrative Graduate Education Research Traineeship, which was an NSF grant. And with this program, we designed a website that's still active at NC State, really just kind of broadly exploring genetic techniques and what, if anything, did they have to offer the field of conservation or conservation on islands. And it really kind of took off from there, skyrocketed. I remember when G-BIRD formed, which is this bottom picture here, words, genetic biocontrol of invasive rodents. And I was in on these talks about, designing this kind of program and the kind of need for assessing this, and is this practical? And G-BIRD kind of kept growing. They had a huge, large DARPA grant. There was a lot of investigation. G-BIRD became a major player in this gene drive community, still is. And then working our way forward here, in 2022, Paul Thomas over at the University of Adelaide, who's part of G-BIRD, created a concept, gene drive mouse, in the lab that I'll talk to you a little bit about. And then moving forward, we're still kind of thinking on this stage. G-BIRD's been a little bit quiet with some changes here. COVID also does things, right? But, what's the future and what are we looking at as far as evolution, alleles? How could this happen? And there's a lot of questions here. So I just kind of want to show you this big old slide that I took from another one here. This is GBIRD, the genetic biocontrol of invasive rodents. Again, you see NC State, Island Conservation on the left here, David Threadgill. You see that the USDA has a major player, Paul Thomas at the University of Adelaide. And wow, what a ride. I never thought when I signed up, I was so excited to get into a PhD program, really kind of what would come out of this. And I don't know if they've tried to kick me out, maybe they have, but I'm still here. I think GBIRD is just awesome to kind of explore. I'm on the steering committee. And it's just been such an interesting program. There's so many different questions to this. And whenever I kind of mention gene drives, the first thought that usually comes to a lot of people's mind is Jurassic Park. And like, nature will find a way. And maybe that's what you all will come up with today. But there's so many questions, right? There's ethics questions, there's regulatory questions. I'm just going to give you my little tiny slice of the pie and mostly focus on my PhD work and some things that I've been working on since then. All right, so this is where, again, I began in 2013, the start of my PhD. I was mostly focused on a meiotic distorter. This is a naturally occurring gene drive known as the T haplotype. And the thought was, could you sex bias a population so that there was not enough females? So rather than putting down toxicants, right, rather than putting an island there, what if you could drive a trait into a local population and then suppress the population because they're not reproducing enough of a particular sex? So the T haplotype is found exclusively in house mice, Mus musculus. And you see kind of a rendition here of what the hope would be is if you created this male biasing, what would happen to a population over time. So a little bit more technical, when it comes to the T, again, we consider it a naturally occurring meiotic distorter. Again, it's found exclusively in house mice. I used a variant of the T called the TW2, which is unique because instead of causing lethality, it causes homozygous sterility, right? So those that inherit two copies are simply sterile. I like to call it a poison antidote system. How it works is that males that are carrying the T, their sperm are weak, right? It makes all sperm weak because mice will mate with multiples, right? But those that carry the T also have the antidote. And so it's able to rescue that. And so when they have the T, it's inherited at rates as high as 95%. So in this TW2. And just to remind you, because it is a weird snowy day, at least here in Raleigh, that a normal chance of inheritance would be 50%, right? And so this is a severe distortion here as high as 95% if they carry the TW2. And you can kind of see this depiction here with the weak swimming and the normal swimming. And then the thought was, what if you could work a male gene into the T? So the T alone is just a naturally occurring, right? It occurs in pretty low levels in populations. It's found near globally, but in small populations. It's like, most places don't have the T. If you can work an SRY gene into the T, this would be the hope here. So what is an SRY gene? Just to remind you, this is sex determining region Y. This is a mammal determining, right? That's going to drive male sexual characteristics. So if you could build the SRY into this T haplotype, then you could sex bias a population to be male. So the SRY gene allows for the initiation of male sex determination. If you inserted the SRY into the T, you could hypothetically drive it into the population. And just again, reminding you here, if XX SRY, then they would be male and sterile. And if they were TSRY males, then they would pass down this construct to the next generation. So this was kind of the hope here. So this became the central research question of my PhD. In deploying a TSRY mouse, a gene drive mouse, right, to remove invasive mice, right, really behaviorally, how wild of a mouse would you need? When I first started this program, it was the very first week of my PhD, and we had started in the summer. And I remember that someone came into the room and they were like, yeah, we're going to take some of these mice. It shouldn't take us that long to construct them, and we're going to dump them on an island. And my work until then had actually been mostly in herpetology. But even then, thinking of a behavioral standpoint, I thought, I don't think that's going to work, right? I just don't think that your lab mouse is going to survive on an island, right? And so that's where I really began this kind of research question of behavior, right? Well, Turns out I needed mice. So at NC State, we didn't have a wild mouse colony. We didn't have an island mouse colony. And so I just finished a yoga class, and it was in about the fall. And my PhD advisor, John Godwin, called me and he said, "Hey, do you want to go to the Farallones?" And I was like, "Oh." We had toured there briefly, just around on a boat. Most people got seasick. But yeah, sure, I'm up for an adventure. And I was like, "Why?" And he's like, "Because we need mice." And so I went out there in the fall of 2013. I actually got stuck there on the island. It's a really one of the highlight stories of my PhD, actually. It's a really cool island. But I was there to collect mice. Why did I go to the Farallon Islands to collect mice? The Farallon Islands have some of the highest mouse densities ever recorded in non-commensal structures. So it's been described like the ground is crawling. There is absolutely overrun with mice. And their plan at the time was to do a rodent eradication with anticoagulants on the island. That is still their plan, is to remove them with the traditional method. They did an environmental impact statement, and they even looked at the aspect of using gene drives, but they thought that that was too far off in the future, and they wanted to remove them now. As of today, or even just yesterday looking, that has not occurred yet, but there are ongoing plans to remove them. So I took some of these mice. We did not capture nearly as many as we hoped for, 21 of them, and got them back to NC State. And then I started breeding them. Before I started breeding them, I had to learn a little bit about mice. Again, my background was actually in herpetology, and I'd never worked with mice until my PhD program. And so it's kind of actually good that I hadn't worked with mice because when I started to work with mice, this is what I was given. These are lab mice. Lab mice are very different, as you could imagine. The mice that we mostly worked with are called black sixes, C57BL6. This is probably the most common laboratory strain throughout the world. And David Threadgill had managed to breed some of that TW2, so not a gene drive with the SRY, but just the gene drive, the TW2, into some of these mice. So they had a black 6 129 background. Again, they weren't transgenic, they just had this naturally occurring T that they were able to breed into it. These are very common lab mice. Probably not the best example of a mouse. So I love this quote here on the bottom left. He's a teenage alcoholic couch potato with a weakened immune system, and he might be a little hard of hearing. For some applications, he's a sorry specimen. And so here I have these Farallon mice that are jumping everywhere. We have people actually quitting because they don't want to work on them because they're used to laboratory mice. And then I have these lab mice that we've taken into a facility, and we're just breeding them. And then again, the hope is maybe I can breed them together. So my ultimate kind of start of my PhD was, now that I have these island mice, right? Will these island mice, and again, I'm breeding just island mice together, so they're island derived, will they even mate with lab mice, right? I'm going to pair them up together. Here's some pictures, right? So on the left, here's a black 6, right? Lab mouse. On the right, here's one of our popcorn Farallon mice, right? And I wanted to answer just some basic questions, right? I wanted to see if sexual selection and isolation has made them reproductively incompatible. I want to understand if there's differences in behavior. Oh, yeah, there are, right? And I wanted to kind of maybe even start to recognize some behaviors and what happens when you take an island population and bring them into the laboratory. And then applied questions. If we wanted to release a island wild mouse, right, would it be able to compete, right? Would it be able to pass down a trait? And so some basic questions and some applied questions here. All right, here's the short story. So these are some hybrid mice, right? So cute little cup, right? So these are some Farallon mice and lab mice, and here you go. This is what they look like, right? And then what came next, right? Then I wanted to see, can these lab strain males reproduce successfully when they're in competition with these island mice, right? And if not, how much back crossing would be necessary to make a competitive mouse, right? So here's my little cute rendition of a competitive mouse, right? If you really wanted to deploy a mouse that has a chance on an island, how behaviorally unique would it need to be? Like, how competitive would it have to be? All right, so this is what I did, and this is going to pain me because I'm going to go through these kind of fast, but this was a long time of my PhD work here. I set up these behavioral arenas, as we called them. These were 3 square meter arenas with melamine to kind of remove scent from. And I would take these island-derived females, right? So island females, and I would have an island-derived male, and then I'd have an opponent male. And I had 10 trials where it was a lab mouse. I had 11 trials where it were these 50%, right? They were hybrids. And then I had 8 trials where they were these 25% lab mouse. And so what did I find? Well, again, just to remind you of the setup and kind of have some more cute pictures here, when I took the lab mouse and a Farallon male and a Farallon female, what I saw, as you can probably predict, is absolutely zero transmission. Lab males do not compete. Lab males are not behaviorally active. It was hard to keep the lab males alive. It was a concern for our IACOC, to be honest. We had to make sure that they weren't getting wounded because mice will fight to the death. So these lab males just had 0% chance. They weren't able to compete. So again, now I have a hybrid. So now I have Farallon females still, right? Now I have the Farallon male, and now I have a 50% wild and lab. And what I found was I had 81% transmission when it was the hybrid. So again, not preferring having the island male, but preferring having a hybrid, right? And so this was kind of startling for us as far as that's interesting, right? And so then I took, in this case, the 25%, right? So now this is an F2 hybrid, right? And I did the same thing where I had, again, Farallon females, Farallon male, and I have this hybrid. And now I have 91% transmission. And so this is really surprising. And so kind of just summarizing here, right? There's no transmission when it's just a simple laboratory male, but when it's a hybrid, they sire over 80% of the pups, right? And so this was really interesting and kind of the heart of my PhD here. And again, it kind of pains me to quickly go through it, but I want to talk about some other things really quickly as well here. And so overall major findings, the hybrid wild lab mice can successfully outcompete, right? We were able to make some super competitive hybrids, right? The greater success of the hybrids was not due to reproductive characteristics. I'm not going to talk about all of those aspects today, but we ran a series of tests. We looked at all different kinds of things. We did not see it was due to anything reproductively. It was linked simply to behavioral differences. However, as you can imagine, being bold might not be the best strategy if you're on an island, right? If these males are super bold, they might stand out. Natural selection might find something else to perch them with, right? So being bold in the lab might be detrimental to being bold in the wild. All right, so again, kind of just reminding you of where I am and where I've been here. And again, I finished my PhD in 2019. So since 2019, there actually has been a construct made in the lab that involves using the T haplotype, right, and a gene drive. So everything I was looking at did not have an SRY gene, right? Here, Paul Thomas at the University of Adelaide had a proof of concept where the T haplotype gene drive mouse, right? The goal here was suppressing gene drive strategy for mice. It spreads faulty copies of a haploinsufficient female fertility gene. And so this is an actual prototype in the lab. I will say though, if you look kind of in the right here, and I can't click quite on my screen here, but what you see here is some modeling of how long does it take for an eradication to occur, right? Think about a traditional method where you're applying rodenticides versus if you were going to release a gene drive, and how long would this actually take to occur in a population? So does it matter if hybrids are more competitive? Probably really yes. When we think about creating a gene drive, there are known fitness costs for carrying a gene drive. We know this. We've seen this in other organisms, right? And so that always the hope has been perhaps you could overcome this detriment by making like a mighty mouse. If you could make this super competitive hybrid mouse, then maybe your gene drive has a chance, right? We know there are fitness costs to carrying a gene drive. Perhaps we can overcome them a bit. So what is needed? If you really wanted to make a gene drive lab mouse and have it be successful, you would have to deploy that mouse into the field, right? You would want a mouse that would integrate into an established population. This is what we call secondary invasion, or what I have coined secondary invasion. And this is some ongoing projects and work that I keep doing when I have the chance here. And it's always been a concern. You have an established population. What happens when you introduce more into a population, right? Also, the other concern, since this is a G-BIRD, genetic biocontrol of invasive rodents, the immediate concern is you would want a way to contain this gene drive. You would want to make sure it doesn't spread elsewhere. You would want a unique mouse that's capable of spreading this gene drive, but you would want it unique to that particular island, since this is what we're focused on here. So locally fixed alleles. And you'd want to make sure you could actually find your gene drive mice, right? You would want to be able to make sure you know where they are. All right, so what I mentioned with locally fixed alleles is this idea of making it unique to an island. So this is some work that has been occurred before and is still ongoing with Kevin O and others that are working on what we call locally fixed alleles. As you can imagine, on the Farallon Islands, drift is probably strong, right? You have a smaller population, right? More prone, especially in smaller population sizes, where drift is likely. And so the thought is drift has caused some alleles to become fixed within the population. And so then what if you could put your genetic construct into that fixed location, right? If this is a unique signature to that island, then even if your mouse was to be taken off the island for who knows why, right, or to have made it off of the island, it wouldn't work in another location because it is working only within that fixed region that's unique to that island. So that's one of the thoughts that G-BIRD has been working on, and you can see some modeling from a paper here. And then how do you find your mouse, right? So actually, I'm super excited to show this because this paper was literally published today. And this paper was a paper I was a part of, and some of my undergrads was a part of, was using environmental DNA to see if we could detect invasive mice. So there was two parts to this project. One was done at the USDA and was a much more laboratory contained. Can we detect them in a laboratory setting under controlled conditions? And over on the right is one of our field sites over by Schenck Forest here, which was what happens actually, right, in a more realistic condition in the Carolina summer heat and humidity. Can you detect mice, right? And I will tell you that accumulation happens fast, as little as one mouse, and you can go ahead and detect DNA when it's in an enclosure. But in the Carolina sun, soil, rain, humidity, the degradation, as you can see here, drops off really fast to very, very low levels. So long story short, how do you detect mice? Well, on an island, you can use things like traps, you can use bait carts. We're arguing this is another tool in your toolkit where you could use environmental DNA, right, to try and monitor your mouse population. And then another paper that came out was looking at could you detect genetic mice? And the short answer is yes. So this is another paper that came out last year over with Tony Piaggio at the USDA. And again, on the left here, what you see is a wild type multiplex. On the right here is this idea of detecting T CRISPR. And again, this was not done outside, this was done in laboratory, but the idea here is yes, the building and the modeling is there where if you were to release a gene drive mouse, you could hopefully hypothetically detect it using eDNA. All right, I want to save plenty of time for questions and hopefully maybe some clarifying because I know I talk fast, but I wanted to save it there. The mice still might win. I know I was tasked, and when I've reached out about evolution-proof strategies, evolution-proof approaches could still fail. We have worked really hard and great, awesome people have done a lot of modeling. And again, there is a concept gene drive mouse, but works in the lab might not work in the field, right? These mice just might not hang. Getting an actual suppression versus eradication. There could be really strong selection against a gene drive mouse. Those fitness costs are there. How hybrid, how competitive. the time for an eradication might be unrealistic if there's ongoing needs, right? The Farallon Islands has plans to use traditional anticoagulant rodenticides, right? How quickly can you build it? How quickly can you make gene drive locally fixed alleles? Is that possible? That's kind of where I land here. So I have lots of acknowledgments. Dr. John Godwin was my PI, kind of the brainchild of all of this with Dr. Fred Gould, who has just been so helpful with the Genetic Engineering Society Center over at NC State. Aaron Shields, who is a current collaborator of ours at the USDA. Dr. Tony Piaggio did a lot of work with the eDNA and has been a big part of GBIRD. Dr. Neil Gimmel helped with funding and support for our project on the eDNA. And I wouldn't be here today without Katrina. So thank you for an awesome invite. And with that, I'll take any and all questions.
Okay, fantastic, Megan. That was really great. And yes, thank you, Katrina, for the recommendation and for introducing Megan. We have time for questions. We have a lot of people in the audience who know many different aspects of this. I'd love to hear some questions from people in the audience. And if not, I can ask some questions to get things started. We would love to hear from others. Yeah, so I was wondering, has this been implemented in any other system, and what other systems is this looked at? I mean, is it looked at in insects for insect pests? And what is is there evidence from other systems that could be brought to bear on this, the system you focused on, and especially I'm especially curious about the evolution proof aspect of this, or the ways that it fails because of selection. Any other insights from other systems related to this?
Absolutely. I mean, this is where it kind of got start, right? Was with insects and that kind of idea of, different techniques. And, it doesn't just have to be a sex bias, it could be some type of sterility. People have worked on suppressing, maybe you've heard of oxytec and mosquitoes. So there have been different systems. The TSRY, again, is an idea unique to mice. The TW2 is unique to mice, but other systems have looked at eradication and suppression. Different people have different feelings also, as well as are you trying to eradicate versus suppress?
Yeah.
That's kind of a trickier one because, it's not as sexy to say we suppressed a population. But in my thought, if it's not causing the ecological problems it was, versus an eradication. Eradication sells more, right? And that sounds better that you completely got rid of something. But I'm not an entomologist. I know that there are other systems where things have been applied. And I know that Target Malaria has done a lot of work trying to suppress and eradicate with mosquitoes and different systems. Wolbachia as well has been looked at.
Yeah, I was wondering about Wolbachia. It's not exactly a gene drive, but I mean, at least I wouldn't consider it, but it has some parallels here.
Definitely. Yes. And I think a
Yeah.
lot of that kind of early start with this project kind of came out of this entomology world.
Uh-huh.
It was actually the Fairlands that got my PI thinking about like, well, what could you apply this to a vertebrate? And it also changes that social discussion, right? You're an insect versus a vertebrate, the social aspect.
Okay, we have a hand up, Cole Butler. Cole, would you like to ask your question?
Yeah, hi, Dr. Sir. Great talk. I just had a question about human assisted dispersal of the mice. I know you mentioned how resistance has been really challenging for eradication efforts, but even if eradication were achieved, mice are small and they fit into tight places. Is human-assisted dispersal, is that a contributing factor, a significantly contributing factor for the mice on these islands?
Absolutely. I mean, that's how they got there, right?
Right.
Like, ships, right? It's a huge concern. And it's an ongoing concern. And, do you spend all this money? Do you spend, millions of dollars to eradicate? And then, yeah, mouse makes it back. Or even worse, you have a rodenticide resistant mouse that makes it there. And now your traditional methods no longer work. And what do you do?
Has this continued to be a problem for control efforts?
Yes.
Okay,
Yes.
thank you.
In some places, Australia, New Zealand in particular, and this is kind of where rodent eradications got started, are better. And you will see signs like, this is an area, or, like, please check your stuff. But it's also hard. A mouse can fit through the hole the size of a dime. So if you can imagine that. Um, they're small, they're tricky.
That just blows my mind. I mean, I mean, I know how hard it is to keep mice out of a house when we lived in New England, and how the tiniest hole they can find their way in.
Well,
That's
I'll also
incredible.
say that on a lot of these islands, rat eradications are the primary concern, right?
Yeah.
The hope with genetic biocontrolled invasive rodents is, it's not just mice, right? I showed you a picture with mouse, but rats are actually more a primary concern. But sometimes they'll do these rat eradications and then they have a mouse problem because rats outcompete mice. And then they realize now
Mm.
we have.
Okay, Fred had a question in the chat. Do you see potential for future use of eDNA for surveillance in the field? Any thoughts
I
on that, Megan?
definitely think it's another tool in the tool chest. Traditionally have been like traps and signs of rodent activity, bait cards, chew cards. I think eDNA is just growing and growing, and the techniques are getting more refined, where if you could even detect a gene drive mouse. I kind of view it more as one of the tools in the tool chest of using it to detect. And hopefully, one of the hopes is you could quantify. Wouldn't it be great to not only just know you have a signal, but how much quantity, right? Do you have like 100 mice versus 1,000 mice? That would be obviously pie in the sky as well.
Okay, and Arvid, thank you for asking a question.
Yeah, that was great. How would you say the appetite for these kind of solutions are these days among kind of the communities that are affected by them? I felt like for 10 years ago, there was this big excitement about these new solutions. And then my impression was that it was a bit of a backlash against it. I'm thinking about that. Was it out in Martha's Vineyard somewhere where they were going to do it and then it didn't happen? And where's the appetite now, and how has that changed over the last few years?
Yeah, that's kind of where I love GBird. And that's my background is I have an interdisciplinary PhD. And, when I started this program, I'm a biologist, but the social aspect is kind of where the rubber hits the road, right? Like you could design a gene drive mouse, and if no one wants it, it's never going to work, right? So one chapter of my PhD is actually looking at social aspects. And gene drives have become more common, or a lot of people have heard the word CRISPR, right? It really depends, on the location, on the social aspect. I don't know if I can put like one finger of this is how the social aspect is, but you're right, Arvid, that there have been huge pushback, and eradication campaigns have failed. Obviously, they can fail because of rodenticide resistance, but actually more often they fail because of social backlash. They either never happen or they're strong. Yes. And so, yeah, there was a case with Anacapa Island, which is also off the coast of California, where, Lord Howe had some social issues, and huge undertaking of trying to explain the need, or the desire. There's also so many aspects that go into it, that, yeah, that's what I really kind of keep me going about GBird. My students and I did a little gene drive survey on campus. And interestingly, what we found in Meredith's community is if a student majored in STEM, they were more likely to say yes to a gene drive. And so we asked lots of questions. It wasn't just about mice. We asked about insects. We just asked about biotechnology. And the interesting thing we found was we didn't find a lot of other trends. But if you were a STEM major, you were more for biotechnology. And I don't know if that's just because of awareness. So sorry to go on a tangent there, but yeah, there's definitely the social aspect. How exactly it stands now, I think depends on by region. I think it depends by awareness. GBird was really having a lot of really good conversations. They're still a player on the scene, but has been a little more quiet lately.
Megan, another social aspect of all of this is in a completely different realm is the social aspect of the mice themselves and their
Yeah.
integration into the existing population. I never thought about that, but it just highlighted to me how important the interdisciplinarity, having people from different fields working together to make this work. How, what were some of the things that you found would help these animals integrate into the natural population? Did you get far enough to have any conclusions on that?
It's really
The
just behaviorally
factors?
driven, you know? I mean,
Yeah.
recording hours and hours of their introgation and like whether they were willing to kind of be bold or shy, right? These
Yeah.
kind of big
So
strategy
personality dimensions, and
dimensions, right?
were there other things you could do to, change the social interactions? I don't know, in terms of distributions of food or how they were, the habitat modifications of one kind or another.
Sure, we played around with like how many to introduce them to. We've also looked at density, and that's actually some of my ongoing work right now, is what we call faking a gene drive, where we're just
Mm.
biasing the sex ratios and kind of seeing how mice behave in a bias ratio to begin with. And then if you
Yeah.
introduce someone, are they able to integrate? Are they not?
Okay. Okay, we have Jason with a hand up. Jason, you want to ask your question? And then I'm going to go to Joe Alcock. Joe, if you'd like to ask your question, here's a chance for you to get ready for that. Go ahead, Jason.
Okay, Megan, I just wanted to thank you. That was a really very, very interesting talk. And I feel like we're almost like long lost siblings. When I was a PhD student, I built outdoor enclosures to study hantavirus infection in deer mice. So I was trapping wild deer mice and founding a colony and doing all of the things you just described. And this is more of a comment. We're as a lab moving into we're arenavirus specialists now. Did you find LCMV in any of your mice? Did you screen for that in your mice that you?
We have, so our original intent was to bring our mice into the biological resources facility, and they were
Yeah.
like, no, right? Because they're
Right.
wild testing. No, we did screen, and no, they did not. But MPV, mouse parvovirus, was always there. And even after breeding and breeding and breeding, our veterinarian wasn't that concerned with it. Just said some populations managed to have it in low levels, but MPV was also something that was there. Yeah.
Because a lot of the questions you're asking, we're thinking about. So like the paradigm for us is you have a hantavirus or you have an arenavirus like LCMV. They're maintained persistently in these mice. They're vertically transmitted in the case of LCMV and wild mus. So we're trying to understand how the virus pulls that off without hurting the host from that angle, but we're also trying to understand possible benefits that actually carrying that virus might convey. And I bet there might be some gene drive type evolutionary events that have happened that allow that virus to stay in those populations. And so this is my pitch to you, that would you be willing to talk offline at some point about this larger
Sure.
set of questions and the things you're doing? I would love to have a few minutes of your time some other time outside of this if we could get that.
Yeah, absolutely. I'll also just say that, one of the things, I mean, this is just me speculating, but, when I say these hybrid mice are much better competitors, it could also just be freeing them from drift, right? Like that,
Yeah.
hybrid vigor, right? Like, it's just freeing them for, you're going to want to have, who you don't want to make same with same, that small little kind of gene pool kind of thing. So, yeah.
Well, wonderful work. Thank you very much. And I'll reach out at some other time. Thanks.
Thanks, Jason. And Megan, I'm going to ask if you would stop sharing the screen just so we can see the whole, or more of the audience at least.
Yeah.
Good. Okay. Thank you. I should have asked that right at the end of your talk. Joe, do you feel like asking your question?
Yeah, sure.
Oh, there you are. Good.
Yeah, I'm here. Thank you, Megan. That's a really, you're doing wonderful work, and it's exciting to see some progress in this area. So I was wondering, it seems like mice pose challenges, in that for a variety of reasons that we've talked about. But I was wondering how these techniques might work for maybe a more slowly reproducing species. I'm sure there are some trade-offs involved with that. I was just thinking about my trip last year to New Zealand, and it seems like there's more societal acceptance to the idea of removing introduced species, at least in those communities. So I wanted to know your thoughts about that.
Yeah, I mean, that it would take longer, right? Like, I mean, even with mice, that takes a really long time with the modeling to reach, eradication. I could see that gene drive taking longer. That's kind of my first thought, but I don't know. I'm open to others. I mean, I guess my first kind of thought is, yeah, if you have more of a slower reproducing species, the generation time is going to take longer. The spread of the gene drive is going to take longer. Yeah, I don't know.
Yeah, me neither. Cool stuff
Okay, and
anyway.
Evan, oh, go ahead, Joe. What's there?
I would just wanted to
Oh.
kind of reiterate, great talk.
Evan, thank you for these comments here. Transmissible vaccine work for loss of virus. Really interesting. If you want to say anything about that, please feel free. And Jay, thank you so much for sharing the report, National Academy's report from 2016.
Yeah, I will say,
Okay. Okay.
going back to a question earlier about like social ability, yes, if you're not aware, like Predator Free 2050 was one of, New Zealand's and Landcare's like big push kind of thing. There definitely is obviously a range of social acceptance and not acceptance when it comes to gene drives. But there's also just a range of, funding and thoughts. And all of this, my thought would need to be trialed in a smaller situation, right? Like the hope was you would go to some type of outdoor pens enclosures where you could contain, right? Maybe a small island where you could actually use rodenticides as a follow-up if your fail-safe kind of worked, right? All of this is just really scale kind of part. And again, I was just such a small part, but also such an exciting part there. And there's lots of people working on this. Like it's not just me. There's people who are looking at the social aspect. I'm part of the ecology aspect with GBird, but there's people still looking at other genetic techniques that could work here too.
Okay, that's great. Evan, go ahead, and this will be our last comment or question. Go ahead, Evan. Thanks for raising your hand.
Yeah, I just looked into it more. So I mean, it's not gene drive, but it's a similar idea. So there's the idea to make transmissible vaccines for a couple of different systems. I know that the Daniel Stryker's group at Glasgow is working on it for rabies virus, and then there's some modeling work at this point for Lassa virus. I looked; it's not LCMV is not the proposed vaccine vector. It's murine cytomegalovirus. But the idea in both of those systems is to take a naturally occurring,
Yep.
highly prevalent virus, right, modify it to express an antigen for a more pathogenic virus. But it has a lot of parallels to what Dr. Sir was talking about, right? I mean, well, first of all, the social acceptance issue now of releasing a transmissible vaccine is probably going to encounter a lot of difficulty. But all these questions about will the recombinant virus be effectively transmitted at the same level as the wild type virus? Can it even invade a population that is already infected with the wild type virus? Those kinds of things. So they're interesting things to think about at this point. I'm not sure how close any of those are to field deployment. But yeah.
Yeah, there was a guy named Stephen St. Joe who was out in Reno who wanted to do this with murine CMV that you could find in Paramiscus species. And the idea was putting hantavirus antigens into that virus and releasing it. And he never got the green light. That when you're dealing with the most abundant mammal on the continent, it raises some questions, right? So, but it could be a very sound idea if you could get it to transmit.
Okay, thanks for sharing that paper, Evan.
Thanks.
Anyone who's interested in it will want to click on that soon because we're going to wrap up here. Thank you very much, Megan, for a great talk. Really great to feature great work from our TriSem colleagues down at NC State and, of course, Meredith College as well, where you are now. So thank you very much for your great presentation, Katrina for the recommendation, and to all of you for coming today. Really great to see everyone. We'll see you at the next one.
Thanks, y'all.